Hi. Good morning. Good morning, everyone. And welcome to the second session of this CISA conference. It was great having you all yesterday with all of you contributing to making this first day a great day of talks, and thanks to all your participation with the questions and with the discussion about different topics. Today we have a really nice day also with plenty of really interesting talks. And I will introduce the plenary speaker in a minute. But we forgot to share with you guys the link to follow live these plenary talks. Yesterday's wasn't possible, but today anyone from outside can watch the talk through the YouTube channel of the Museo Nacional de Ciencias Naturales. You just have to write down Museo Nacional de Ciencias Naturales YouTube, and there's a in directo, live, and you can follow Michelle's talk in a minute. Same for tomorrow's talk by Isabel San Martin. And good thing is that if you miss that, you can watch it again because it will be stored in this YouTube channel. There's something else that I want just to mention, which is that we need the presentations to be uploaded for the part after the coffee break. We have all of them for this part just before the coffee break, but as soon as you can, please come here just before the coffee break and just upload your presentations. So, Anna, please.
Thank you. Yes, upload the presentation because Carlota is having a heart attack, okay? And she needs a coffee as well. It is an honor to present Michelle Leger, who is a truly evolutionary biologist with a fantastic portfolio of research in the evolution of protists. She got her PhD in Dalhousie University in Canada, yay, Canada. And then she decided to move to warmer latitudes, very wise move, to Barcelona, where she's been in the lab of Iñaki Ruiz Trillo. Where she's studied the origin and evolution of protists with different approaches, from the biodiversity all the way down to the functional genomics of these guys. She's a very cool scientist, and I hope you follow her talk today and her work afterwards. Thank you very much. La presentación, ¿y dónde voy? Estoy underprepared. ¿Así?
Thank you very much. Can you all hear me right? That's working? Okay, so first of all, thank you so much, Anna and Sergi, for the invitation. It really is such an honor to be here among you, sharing my work and listening to the fantastic talks these few days. So as Anna mentioned, I'm a postdoc in the laboratory of Iñaki Ruiz Trillo in the Multicell Genome Lab, and I'm going to be talking to you about some of the work that I'm doing about the evolution of protists closely related to animals, and I hope to spark a general interest in you on the diversity of protists and their importance perhaps to the organisms that you study as well. So here are a couple of pictures of animals and plants that have been talked about during this Congress. On this slide, I'm also showing close to animals a picture of some fungi, Saccharomyces, and the reason that I'm doing this is to show you that most of the knowledge that we have of basic cellular molecular biology comes from model organisms in just 3 groups of eukaryotes. So fungi with models such as Saccharomyces, animals with models such as Drosophila or mouse, and plants with models such as Arabidopsis. But in fact, these represent just a small part of the full diversity of eukaryotes. And this is actually a point that was made really nicely yesterday on the talk on testinated amoebae. This slide, of course, has a typo immediately. I'm seeing this, one of these groups should be labeled SAR, not Haptesta, my mistake. But what I would like you to take away from this slide is that animals, plants, and fungi represent just a tiny part of eukaryotic diversity. The diversity of eukaryotes as a whole is currently classified into a number of supergroups that are shown here in shaded triangles. But there are many more lineages even outside these supergroups that are of uncertain placement. And what I'd like you to remember about this slide isn't the specific names, but there are very many eukaryotic lineages that together represent an enormous morphological and ecological diversity. So on this slide, I've shown some of the morphologies that the eukaryotes outside plants, animals, and fungi, collectively known as protists, can take. They have many different roles in the environments. They can be predators or photosynthetic organisms. They can include microalgae, and the majority of these organisms are microbial. So there's an enormous unseen diversity of eukaryotes that we don't fully appreciate. And again, just to show you the position of animals and fungi together are just part of this one small lineage within just one supergroup of eukaryotes. So this is the diversity of eukaryotes that's out there that we're barely even touching on in this meeting. And why do we need to understand this eukaryotic diversity? And I'd like to make the case to you that even if you don't personally study protists, they do affect every aspect of your lives and the lives of the organisms that you currently study, because lesser studied eukaryotes are are parasites of medical and agricultural importance. This is an organism that caused a major famine that affected human history in Ireland in the 19th century. They may be symbionts of animals and other organisms. So this is a symbiont of corals that's implicated in coral bleaching. They may be major predators. So this nondescript organism is actually numerically an extremely important predator of microbes in the ocean, affecting oceanic ecosystems. Their exoskeletons form structures that we see with our eyes in the world around us. So cliffs can be formed of the exoskeletons of microalgae. And they're major oxygen producers in global oceans, producing up at least 40% of global oxygen production. So these have major effects on all global ecosystems, on human health, on agricultural systems, and their importance in the world is changing and becoming more important as climate change progresses. So this is an example in which the so-called brain-eating amoeba, Nigularia, has a range that is spreading, infecting humans at more and more northern latitudes as climate change progresses. And so we really need to understand the basic biology of protists. Even though protists aren't a single group of organisms, they are, again, very diverse and represent several major groups of eukaryotes. So from all of this diversity, I hope that I have sparked your interest in going to read more about this, but I'm still going to go back, narrow down my focus back to this one small group of animals and organisms that are closely related to animals, partly because this is because the majority of people presenting here study animals, and partly because this is the major topic of the lab that I'm currently doing my postdoc in. So what makes an animal an animal? At the top left here, I'm showing some examples of animals, and what all of them have in common, from the nice people who attend CISA meetings to bears to sponges, is that they share certain features, namely embryonic development and spatial cell differentiation, which means that different cells within the organism take on different functions at the same time, depending on their location in the organism. And this is made possible by what we in the multicellular genome lab tend to refer to colloquially as a multicellularity toolkit, namely a set of proteins that help cells to adhere to one another, to adhere between cells involved in cell-cell signaling, so that cells can communicate with each other and sense their environment and respond as the result. And major regulators of all of these functions, as well as a program of transcription factors that together regulate complex behaviors of individual cells and cells within the organism as a whole. And so all of these proteins that form the multicellular toolkit were present in the last common ancestor of animals, shown here with a little red star. But in fact, work in the multicellular genome lab in past years has shown that many of the proteins that carry out some of these essential multicellular functions were in fact present not just in the last common ancestor of animals, but in fact in much earlier ancestors. So some of these proteins were actually found to be present in protists that are closely related to animals, and as a result, were likely present in shared ancestors of those protists with animals. So this is a topic of major interest in the multicellular genome lab, to try and identify proteins that are essential for a multicellular lifestyle in animals, to try and understand which of them were present before. Or I can shout. No, that's better, thanks. And to try and understand which of them might have, in a sense, potentiated the emergence of animals, which one of them might have been functionally co-opted, that means acquired new functions around the origin of animals. Sorry. And so I'm going to start by telling you a little bit about the organisms that I'm going to be discussing in more detail. The general name for the grouping of animals up here, fungi down here, and the closest relatives of animals and fungi collectively is a Pithecanthra. That's a term that I think most of you will be familiar with already. And more narrowly, the grouping of animals and their closest protist and relatives to the exclusion of fungi and closely related lineages is Holozoa. And collectively, several different lineages shown on this diagram are the organisms that I'm going to be collectively referring to as the holozoan protists. So these are all microbial lineages that molecular data have shown us are likely closely related to animals. And these organisms, which are far, far, far from all of protist and diversity, but which are just a very tiny, narrow part of protist and diversity, are nevertheless themselves also quite morphologically and ecologically diverse. So just to mention just to introduce some of these major groups, the closest sister groups, this is the group that's most numerically described. So these are organisms that are, again, single-celled. They're bacteriivores. They're found mostly in aquatic environments all over the planet. And they have over 360 described species. And they're able to form rosette-like colonies. So upon induction by environmental cues, such as the presence of prey bacteria, these organisms start to divide without fully separating, and they form clonal colonies, so a simple form of multicellularity. The second sister group shown here is Phyllosteria, and there's no major synapomorphy of this clade. We only know of 3 genera so far. Capsospora, which is an endosymbiont of a freshwater snail, the Bionfilaria, the host of Schistosoma. And 2 free-living aquatic groups, Ministeria, which is marine, and Pygoraptor, which has 2 freshwater species representatives. Both of these organisms we believe to be free-living predators, but we know very little about this group. We do have a study Capsospora in the lab, and our studies there have shown that Capsospora doesn't form clonal colonies, but under shaking or the presence of certain nutrients, it does form aggregates, so individual cells coming together and forming simple multicellular structures. A third group is the Corellicotria, which we only have 2 known genera, and 2 known species indeed. Both of which were isolated from, one of which was isolated from a marine environment, one from a freshwater environment. Cisamonas is a eukaryovore, and Corellicotrium we assume to be some kind of heterotroph, but to be honest, we have very little knowledge of its basic behavior in the environment. And Cisamonas has been described as forming clonal colonies and aggregates under laboratory conditions. And finally, the Ichthyosporea are a group that might interest you in particular because these are often parasites or endobionts of animals. We know of 120 described species, so they're a little bit more abundant than Phyllosteria or Corellicotria. And they're generally parasites or associated with marine invertebrates or fish, sometimes amphibians, in one case humans. There have been reports of free-living species, but we haven't been able to confirm that these species are indeed free-living and not simply a free-living stage of an organism that's normally parasitic. So of all of these 4 major groups of lineages closely related to Metazoa, I'd like to make the points that Holozoan protists have very diverse morphologies and lifestyles. So all of these groups collectively represent different morphologies, but also parasitism, commensal lifestyles, they're eukaryovores or bacteriivores. Many have simple multicellular life stages, and that's something that's of great interest to us, studying the origins of animal multicellularity, because we're trying to understand whether any of these multicellular toolkit genes present in some of these organisms also help them to have these simple multicellular stages. And finally, Holozoan protists have a taxon sampling problem. So for 2 of these groups, we have a reasonable number of representatives, but at least 2 of them, we only have 2 to 3 described genera. So some of you were mentioning yesterday that you had casually sampled 400 or 250 new species of your group of interest. Imagine if you only knew of 2. So this is the problem that we're up against. Furthermore, as a result of this low taxon sampling in some groups, taxonomic relationships within the Holozoa are disputed. So different studies have come out over the years that have reached different basic topologies of the backbone of Holozoa. I mentioned Corellicotria and Ichthyosporea, and these are 2 groups that sometimes form a sister relationship in some studies, together forming a group named Teratosporea. But in some other phylogenies, Corellicotria is the sister to the rest of Holozoa, and in yet others, Ichthyosporea is the sister group. And of course, part of the issue here is that this group, Corellicatry, only has 2 representatives, and they are fairly long branching, so no wonder that we have this problem. But it's something that's of interest to us to try and clarify this, because without understanding the taxonomic relationships between holozoa, we can't fully understand the order in which different proteins evolved along this lineage. And complicating the story is a small organism that was isolated a couple of years ago by a different group named Tunicaraptor unicontum. And in the work of the group that published the description of this organism, it took on pretty much any position in holozoa that you can imagine, and also ended up challenging relationships within a holozoa as a whole. So what we'd like to do in our lab is fundamentally understand what are the relationships within holozoa. We can then use that information together with understanding what traits holozoan protists have, and put together this information to try and understand what features might have facilitated the emergence of animals, partly by understanding which features of animals were present before animals as a whole and which ones were animal innovations. And so helping us in our quest recently is some work that I'm going to be talking to you about together with collaborators in the United Kingdom, David Bath at the Natural History Museum and his then student Ander Urrutia. And they were searching for putative protists and parasites of amphipods. They had a species named Echinogammarus, and they found that some of these organisms were infected by an agent that affected the motility and behavior of the pods. So infected individuals had poor motility. You can see that this organism looks quite sad here. And logical investigations revealed the presence of some granulomas, suggesting perhaps that the organism was mounting a molecular defense against a parasitic infection close to the site of some rounded cells that were the putative parasite. So this is actually an important point to make, because one of the problems that we have in protistology is knowing what is a parasite and what is just present in an organism as either a commensal or part of the, or it might be passing through the intestinal tract. And this kind of phenotype and molecular response is what allows us to believe that this is indeed a parasite. So based on 18S sequences, Ander and David believed that this was a new holozoan. And because it had only been found in May and it was a small spore, they named it Chiquispora philomaios meaning loving of May. And they found that it formed rounded cells within muscle tissues of the host. And here at the bottom are some pictures showing the TM morphology of the organism. And so we tried to generate data from this organism to examine its gene content and phylogenomic position. And one of the problems that we've had in general in protistology is trying to get good data from mixed samples. So many protists are either parasites that have to be isolated from a host, or they're grown together in culture with prey eukaryotes or bacteria or both. And so it's actually been a major challenge to generate high-quality data from protists, given that we often can't isolate them into pure culture. And metagenomic binning has been a major boon for research in these questions. So just in the past couple of years, we've been trying to apply metagenomic binning to eukaryotic protists. And what this involves is taking a mixed sample, sequencing its genomic DNA, and then assembling it into contigs, and finally separating the contigs into different virtual bins based on tetranucleotide frequency. So the frequency is any given set of 4 nucleotides in a row, and on read coverage. And this has allowed people working in the field to generate bins that supposedly include only contigs from each given organism that's in the sample. And this had never been tried on a holozoan protist. And in fact, at the time that my collaborator, Konstantina Mitzi, who some of you know, started this project, it hadn't actually been done in a eukaryote, in a protist at all. And so we tried this technique on the sample from Chiquispora together with its host. 38% of the contigs were assigned to bins. The remainder could not be concretely established. And the majority of these, we assigned taxonomy to each of the organisms represented by each of these bins. And we found that the majority were likely bacterial organisms. And of the remainder, the bins that were predicted to be eukaryotic, only 2 eukaryotic bins had high Busco genome completeness. So this is a score, Busco is just a score of genome completeness overall. And based on 18S searches, we believed that these 2 complete bins were representing Chiquispora and its host. The remainder might be trace food sources, perhaps, from food that the host had consumed. And we were quite pleased because the completeness within this Chiquispora genome bin was actually comparable to the Busco completeness of genomes that we've in the past obtained from pure cultures. So this was something that was really great to see. In addition, we had no duplicated Buscos in that bin, and we extensively tried to examine, to search for potential contaminants in that bin. We were very concerned that we might have host sequences or bacterial sequences in it. And in fact, we only identified 2 contigs that were putative contaminants from the host, so, and a couple of other ones that were possible yeast contaminants. So we were actually quite happy, not just for the completeness, but also the relative purity of this bin. And so our first order was to try and place Chiquispora more concretely within Holozoa. We combined, we worked together with Ramon Massana at the Institute of Marine Barcelona and his student, his postdoc, David Escador, because they had together found generated some single-cell amplified genomes from marine water off the coast of Blanes near Barcelona. And they had identified a putative marine small holozoan or mass hole that they also wanted to place. And so we decided to generate some phylogenomic data for both this and Chiquispora together. So we used 255 concatenated marker genes based on Busco analyses. We, in general, in the community of people looking at phylogenomics of protists at deeper levels, we tend to use concatenated data sets rather than consensus tree building, because the latter is more vulnerable to long branch attraction artifacts, which is a major problem for us. Jesus Lozano in his talk yesterday mentioned the CAT-PMSF approximation of CAT models, and we also decided to use that for our data set because it's better at modeling differences in the exchangeability rates of different amino acids in a data set. And shown on this tree are non-parametric bootstrap support values, so these are real bootstrap values. And we found that a grouping together of Ichthyosporia and Corallocytria, which I mentioned earlier as one of the possible topologies in Holozoa, had a support of 100%, so Pterotosporia lives. We were very interested to find that Chiquispora and the new marine small holozoan named E11 were forming a clade with full bootstrap support again, together with this mysterious marine organism Tuniciraptor that had been messing up the tree of Holozoa. So this does seem to form a new little clade. And this clade seemed to branch sister to existing Philisterian or known Philisterian sequences. So the bootstrap support there is 79. It's a little bit borderline, but we're currently running fast site removal analyses to to see whether this position is robust to fast site removal. Fundamentally, we're classifying this group of Cichespora, Tunicaraptor, and E11 as a new philosterian clade. And all of these putative philosterians, animals and choanoflagellates together are form the clade Phylozoa, and that again has good bootstrap support in our phylogenies. So Cichespora, Tunicaraptor, and E11 represent a new holozoan clade. They're probably philosterians, and what's great about that is that they would almost double the number of philosterian species known, which helps us in our analyses. And furthermore, since Cichespora is a parasite, that's a lifestyle that hadn't yet been described in the grouping of Philosteria, allowing us to examine the effects of different types of lifestyles within this group of protists. So Cichespora is a parasite, and I mentioned earlier that Capsospora, one other member of Philosteria, is an endobiont, and these would be to the exclusion of free-living members of this clade. So one of the questions that we started by asking is, what can Cichespora tell us about endobiotic lifestyles in the group? Excuse me. Or do we see any consistent losses of pathways in the group that might indicate that Capsospora and Cichespora have adapted to a parasitic lifestyle by losing similar metabolic genes? So in protist parasites, we often see, or endobionts, we often see losses of specific metabolic pathways because the organism in question can derive specific nutrients from the host. And we found that actually they seem quite different. Capsospora is notable for having a complete loss of ubiquinone biosynthesis, which is an essential to function of the electron transport chain. Presumably it derives this from the host instead. And Cichespora has quite a lot more losses metabolically. I'm showing the Busco scores at the top here so that you can get a sense that probably these losses are real and not just a function of low coverage. And specific things of interest that it had lost are biotin biosynthesis, which is a pathway that's convergently also been lost in Trypanosoma, a very distantly related protist parasite, and glycogen biosynthesis and degradation, which are pathways that have been found to be consistently lost in bacteria that are endobionts, whether parasitic or not, and in microsporidium and parasites as well. So Cichespora seems to have independently adapted to parasitism by loss of specific pathways that are convergently also lost in different protist parasites, but its patterns of losses are not similar to the endobiont Capsospora. What can Cichespora tell us about the animal multicellularity toolkit and how that might have evolved? So we first looked at cell-cell adhesion, extracellular matrix adhesion, proteins, signaling proteins, regulators of the cytoskeleton, signal transducers, and transcription factors, which are elements that have previously been described as being either animal-specific or more widely present in holozoa. And you don't need to remember the names of any of these proteins, but the point that I just wanted to make was that as a whole, we found that Cichespora was fairly consistent in its molecular machinery with other holozoans and especially other members of the Philosteria. It had a fewer components of this than Capsospora, but what we were interested to find was two particular genes that had not previously been described outside of animals. We also found these genes encoded in the Tunicaraptor transcriptome, which was great because this helped to cement the idea that these weren't partial animal contaminants. And both of these proteins were regulators of, in one case, calcium stores, and in the other case, broad-spectrum protease inhibitor. And while both of these proteins have fairly narrow molecular functions, they're of importance to the animal lifestyle because both of them act as regulators of apoptosis, and in the case of the first one, MZB1, of diversification of B cells in the immune system. So both of these elements that were later co-opted into functioning in the regulation of apoptosis and immune responses were also present here in the shared common ancestor of Philisteria, Metazoa, and Chronoflagellatea much earlier than previously believed. And the only point I really want you to take away from this slide is that every time that we find a new holozoan protist, we find a few more of these so-called animal-specific genes. It helps us to identify an earlier origin for something that's previously believed to be essential to animals. So again, taxon sampling matters. We need more of these holozoan protists. And the fact that I mentioned apoptosis brings me to the final part of my talk, which is on the evolution of regulated cell death pathways. And this is an essential function of animals that interests me because it is so essential to animals functioning in their everyday life and in their development. So regulated cell death is an overall term that encompasses a wide set of processes that lead to death of specific cells in a genetically controlled, regulated manner as a result of either external or internal triggers. And within this larger term, a more narrow term is programmed cell death, which is a set of processes that's involved specifically in developmental contexts or homeostasis. And these are essential processes in either development of animals, shown by some of these pictures here, or as for the everyday turnover of cells in a multicellular organism. And the term that you'll probably be most familiar, very complex schematic, is that apoptosis is a set of regulated cell death processes that's classified as being dependent on caspases, a specific family of protein cysteine proteases, and that this apoptotic pathways involve specific proteins and domains that are involved in recruiting, activating, or blocking the actions of caspases. And all of these regulated cell death processes are in contrast to accidental cell death, which is catastrophic and not genetically regulated. So what do we find of regulated cell death proteins outside of animals? And to try and investigate this question, I started by conducting sequence similarity searches, building custom profile hidden Markov models for more remote homology assessment. And in some specific cases, I used AlphaFold to build protein structure predictions of candidate proteins of interest in some holozoans. And I used those proteins for structure searches against public databases of protein structures to try and identify and confirm the identity of sequences that had very low sequences, so that were very poorly conserved and had low sequence similarity. And I found that the majority of elements in apoptotic pathways are indeed animal-specific. So in this figure here, this part of the schematic shows caspases and the domains that are involved in their activation and recruitment. So I found that everything in gray here in this figure only seemed to be present in animals. However, some domains that have been linked to blocking the activation, to blocking the action of caspases, so inhibitors of apoptosis seem to be much more widely present across the pisticonta as a whole. Some upstream regulators, inducers of regulated cell death, such as P53 or NF-kappa B, which have functions in cell death differentiation and immune responses, are more widely present across holozoa as a whole, as are some downstream effectors, such as apoptotic chromatin, condensation protein. And most interestingly, some proteins known as the tumor necrosis factor proteins that are involved in triggering regulated cell death are found, have some elements that are found widely across eukaryotes, but most interestingly, the main trigger of this pathway, tumor necrosis factor itself, which had previously not been described outside animals, was in fact broadly present in cranoflagellates. And what was interesting to me when I searched to try and identify the distribution of these proteins was that these proteins were found in bacteria, cranoflagellates, and animals, and most specifically in some species of bacteria that form simple multicellular structures. So these are proteins that are found in animals with complex multicellularity, in cranoflagellates that can form simple clonal multicellularity, and in simple multicellularity producing bacteria, which I'm just going to flip through those slides because I think I'm a little bit low on time. But basically, just to recap, that that many elements that are of multicellularity toolkit genes were present broadly outside animals, but that some of them remain animal-specific. That taxonomic sampling is important to try and understand which ones really were animal-specific. And what I want you to take away more broadly from my talk is that the vast majority of eukaryotic life is outside animals, plants, and fungi. Get interested in them because they do affect all of your organisms as well. Understanding this diversity doesn't just help us understand modern ecosystems, but also major evolutionary transitions. Chicospora, Tunicaraptor, and E11 are three holozone protists that form a clade that nearly doubles the number of known phyllosterians, so a major group of holozone protists. And an endobiotic lifestyle emerged at least 3 times in holozoa, in Ichthyosporea, Capsospora, and Chicospora. And finally, that animals are caspases, the major regulators of apoptosis in animals, are indeed animal-specific, but that some of the pro-apoptotic signaling proteins that work together with and trigger these proteins actually did predate animals. And and some open questions that we still have are what explains the numerical disparity between different holozone groups? Why are some of them apparently so successful and abundant, relatively speaking, and others only have a couple of known members? How common are holozone protists really? So this is a tree shamelessly stolen from Rosa that shows with red stars lineages in which holozone endobionts or parasites have been found, but we're generally interested about which other ones might also have some. Look at your data if you have a lot of data, or look at your samples, see if you would like to search for holozone protists or collaborate with us in searching for holozone protists. And one of the questions that I have that I'd like to explore is whether these multicellular toolkit genes also facilitate an endobiotic lifestyle in holozone protists. So do any of these signaling and adhesion proteins actually allow organisms such as Iberians to interact with their hosts? So I would like to start by thanking my advisor, Iñaki Ruiz and all of the members of the lab shown here who have been collaborators on this work, my funders, the CISA supercomputing group that allowed this work, and especially Anna, Sergi, and everyone else who has been involved in this amazing meeting. Thank you.
Can you hear me? No?
There was a hand up there.
Hola, hola.
I see a very eager person to ask a question.
Hi there. Thank you. Fantastic talk. Sorry, I missed the very, very beginning. I have a couple questions. So first question, you said you're using a concatenated data matrix to infer the phylogeny because it accounts for long branch attraction, but it is well known that a two-step multi-species coalescent approach actually deals better with long branch attraction. I'm surprised you're not checking that you're not building gene trees and then from those gene trees building species trees of these organisms.
So the literature that I've read suggests that coalescent approaches are more vulnerable to long branch attraction problems for deeper evolutionary events.
Okay,
but I would like to talk to you about that.
Thank you. And then the other thing is, it is well known, for instance, in fungi that horizontal gene transfer is sort of rampant, and plants do the same, and anything outside, your entire diversity of protists also do the same. So I'm guessing these protists here also have. So that's why I was going for why don't you build gene trees to see if you can actually identify instances of horizontal or lateral gene transfer.
We did build signal gene trees for all of these markers, so precisely for that reason. And in fact, we found that some of the data sets that had previously been used to investigate these questions had what I believe to be either lateral gene transfer or contaminants, or in a couple of cases, paralogs. So we think that perhaps those instances might have contributed to some of the confusion going on about the taxonomic relationships, but we did use the single gene trees to identify them and remove them from the final data set.
Thank you very much.
Thank you.
Okay. Hi, Michelle. Excellent talk and excellent the things that you are doing. I do.
Sorry, just to make sure that we have fairly complete genomes when we do our phylogenomic analysis.
Are you getting like to the level of genome completeness of some people that are doing chromosome scale assemblies of animals? I don't know. I guess that you are not at this point right now, but.
Well, actually, we are. So a couple of people recently, so I think somebody mentioned the synteny analysis paper yesterday, that was Rosa. And that paper actually published some chromosome level analyses of the genomes of 3 different holozoan species. But at the moment, we have, we don't have that many species for which chromosome level assemblies are available. The best that we can do is, but that is something that we're exploring for the future. We are still quite happy with the coverage and the gene content taxa you can see on the bottom there. And in those cases, what our lab is trying to do is get nanopore sequencing going for each of these organisms. So when we're doing analyses to try and place the phylogenomic analysis, for those organisms, we tend to use transcriptomic data rather than genome data, just because it has much better coverage. But in any case, every new organism that we try and sequence within this group requires a period of trying to optimize the sequencing protocol.
Thank you again for this very interesting talk and for taking us out of the animal and very little plant world that we're so focused on. You mentioned that in many instances you don't have synapomorphism, and I guess it's morphological synapomorphism. What did you use instead? Did you use genome content synapomorphism, or?
We literally just placement in phylogenies, and this is, in some cases, there are synapomorphies, or either morphological or molecular for different groups of protists, but in a couple of instances, such as phylogerians, especially with low numbers, we don't have anything particularly concrete so far. We're always on the lookout for, but we tend to rely on phylogenetic placement rather than any characters.
Any more questions?
Thank you, Michelle. Very interesting talk. Regarding the pathways that you are not able to detect in some of your lineages or species of interest, have you tried to go the other way around, try to first annotate functionally, try to find convergence, and then see if they may be doing the same thing but with a completely different machinery? Because that could be interesting as well.
Yes, so not specifically by function. We did also find analyses to try and identify any particular patterns of absence that were fairly consistent between, say, all of the parasitic or endobiont lineages within holozoa. To be honest, we didn't, or at least I didn't particularly expect that many convergences, because that's not something that we see across protists as a whole. It does seem to be that whenever we see a new parasitic lineage in protists, there are many pathways, many paths to becoming a parasite, many different metabolic pathways that can be lost. So sometimes there are convergences, but there's not a shared pattern of losses or even lateral gains by lateral transfer that makes a parasite. There's not like a parasitic toolkit.
Thank you again, Michelle. And I guess it's time for the next session, and a big round of applause.